Hi, my name is Nabia Chaudhry, and I have been working in Dr. Chan's lab this summer. My research project focuses on exploring the impact of niche cells on regulating muscle stem cell function for treating muscular dystrophies. When our muscles are damaged, the muscle stem cells proliferate, differentiate, and eventually become myofibers. Not all muscle stem cells do this, however. Some of them go back into the quiescent phase following proliferation. This is so that there is a reserve of cells so that when there's future muscle injuries, there can be muscle stem cells that can jump right in. Unfortunately, there are certain diseases in which this regenerative process is exacerbated. This is when the muscle stem cells get exhausted, and this can have certain conditions such as cardiomyopathy along with respiratory problems. Transplanting donor muscle stem cells into these patients seems to be a promising option for treatment. However, we need to ensure that there is long-term effectiveness of this treatment so that the muscle stem cells can retain their functionality. The regeneration process is regulated by both intrinsic and extrinsic factors. This is where fibrodipogenic progenitors comes in, or FAPs. In my project, we aimed to see how FAPs regulate muscle stem cells' self-renewal ability. The results showed that when muscle stem cells were co-cultured with FAPs and then allowed to grow in a differentiation medium, there was a much higher percentage of muscle stem cells as compared to myogenic progenitors. This was in high contrast to the control, which had equal proportions of both muscle stem cells and myogenic progenitors. Understanding the role of FAPs in regulating muscle stem cell functionality is very important for advancing muscle stem cell therapies to treat muscular dystrophies.
Hi, my name is Sierra Chen, and this summer I'm working in the Visible Heart Labs, which is headed by Dr. Paul Izio. My project is focused on the anatomy of the coronary arteries, which wrap around the heart and supply blood to the heart muscle. However, in coronary artery disease, which is the leading cause of mortality in the United States, these arteries can become clogged or even completely blocked. Nowadays, coronary artery disease can be treated by placing a catheter through a tiny incision in the arm or the leg and feeding it all the way up to the coronary arteries and opening the blockage. However, one challenge with these procedures is that the anatomy of the coronary arteries varies significantly between person to person. So when designing medical devices that are used in these procedures, we must take all these variations into consideration to make sure that these devices work for everyone's unique anatomy. For my project this summer, I'm going to be comparing the anatomies of the coronary arteries from people of different ages and genders. Using high-quality CT scans of human hearts, I'm creating 3D computerized models of these coronary arteries and measuring their lengths, diameters, and so on. Our goal is to use this research to ensure that these medical devices will be effective for treating coronary artery disease in all patients.
Hi, my name is Anita, and I have been working in the Comdar lab this past summer. Our lab focuses on research regarding Duchenne muscular dystrophy, which is a genetic disorder caused by alterations in the protein dystrophin, which ultimately leads to progressive muscle degeneration and degradation. Dystrophin is a very important and critical protein for muscle cells because it helps stabilize the muscle fibers against muscle contraction and stretch. And another one of these proteins is something called A7B1 integrin, which is a receptor that spans the membrane of these muscle cells. And previous research have noticed that in skeletal cells, A7B1 integrin plays a role in kind of balancing out the loss of dystrophin, but its role in cardiomyocytes has yet to fully be examined. And that's where my project kind of helps with this next step. I evaluated the expression of A7B1 integrin in 3D bioprinted cardiomyocyte models of DMD. 3D models take advantage of the differentiability of stem cells while also providing a more and accurate representation of the interconnectedness of cardiomyocytes that you might see in normal human heart tissue. Understanding this protein better kind of gives us another view of another potential therapeutic target that may be used for those struggling with this disorder.
Hi, my name is Aish Gujral. I'm an incoming sophomore at the George Washington University Guaranteed Medical School program. And this year, as a Lillehei Heart Institute scholar, I worked with the DDT lab and the Minnesota Muscle Lab. I worked as a small subsection of a larger NIH grant for muscle diseases, also known as myopathies. You may already know that muscles Contractions is usually regulated by calcium. Within muscle cells, there's two pumps that deal with this: ryanodine receptors that release calcium and Circa pumps that take in calcium. And oftentimes a lot of these myopathies result from dysregulation of these intracellular levels of calcium, essentially calcium going in and out at the wrong amounts or at the wrong time. So my research project is essentially about screening thousands of different compounds that are known to have some effect, and then narrowing them down into 10 or 15 different compounds, and then from there we can analyze their substructures and then potentially create drugs that can have therapeutic uses to solve problems related to these myopathies in muscle cells.
Hi, I'm Ellen, and I'm working in the Perlingero lab this summer. I'm investigating a phenomenon in skeletal muscle regeneration. Right now, there are a variety of muscular dystrophy diseases that skeletal muscular regeneration therapies can help with. My lab is taking a cell-based approach to treatment. The anomaly my lab has previously discovered is where embryonic-like stem cells that my lab used are functioning like adult stem cells when transplanted into mice skeletal muscle. These cells contribute better overall to muscle regeneration than regular non-adult stem cells do. I'm studying a protein called periostin, which is suspected to play a significant role in this maturation process because it is only expressed by these cells after they are transplanted into the mice. I want to simulate and optimize periostin expression in pluripotent stem cells without needing to engraft them into mice muscle. This will save time and resources so that we can use my findings in future periostin studies and understand the role of periostin in skeletal muscle regeneration.
Hi, my name is Sahil, and I'm in the Townsend lab. We study Duchenne muscular dystrophy, which is a progressive and degenerative disease caused by mutations in the gene for dystrophin. Dystrophin is the core component in the dystrophin glycoprotein complex, a group of proteins that associates the outer membrane of muscle cells. In the lab, we are interested in how the heart responds to injury. My project focuses on the protein synthesis in dystrophic and non-dystrophic mice following injury. Differences in protein synthesis after injury may explain how non-dystrophic heart muscle can repair itself while dystrophic heart muscle cannot. To measure the role of protein synthesis, we injected mice with a stimulant to increase their heart rate and cardiac contractility. 3 days later, we injected mice with pyrimycin, a toxin that stops protein synthesis at low doses. After we injected these mice with pyrimycin, we harvested their heart. We used fluorescently labeled antibodies to select for pyrimycin within the harvested hearts. Our preliminary analysis reveals that hearts with more injury have greater levels of protein synthesis.
Hello everyone, so my name is Shelby Olson, and this summer I had the amazing opportunity to work with Professor Ogle and Dr. Singh in developing a better understanding of the early mechanistic changes involved in hypertrophic cardiomyopathy. In their most recent study, they determined that there was some sort of altered connection between the sarcomere and the extracellular matrix. We know this because the most common mutation associated with HCM lies in the sarcomere, and as the disease progresses, it causes changes in the composition of the extracellular matrix. Our goal was to determine different protein pathways involved in the facilitation of these changes. We did this by culturing 3 sets of stem cells, one with the mutation, one without, and a control line. We then used qPCR on days 15 and 30 to quantify the levels of mRNA expression associated with the 11 proteins we chose to study. The results of this experiment will help provide future therapeutic avenues for delaying disease progression or even preventing onset altogether.
Hi, my name is Benjamin, and this summer I worked in the LHI lab where we focus on the cellular mechanisms behind heart attacks. Normally in the cells, a certain amount of calcium is required for the mitochondria to produce energy. but too much calcium in a heart attack can also cause cellular damage and tissue stress. We wanted to see if we could prevent this stress by using calcium biosensors, which normally just measure the amount of calcium in the cell. Out of 3 different kinds of biosensor proteins with different binding strengths, the one that bound to calcium weakly was best able to prevent apoptosis, a kind of stress-triggered cell death. This was because it only bound to calcium at very high calcium levels, and so it didn't get used up at low resting levels. That made it able to protect against calcium stress in a human heart cell line. The next step is to apply these tools in an animal heart disease model to see if they can prevent heart attacks there.
Hi, my name's Sneha and I'm working in the Van Berlo lab this summer. In response to increased stress like a heart attack, the heart undergoes a remodeling process, an attempt to prevent a loss of function. One of the changes that occurs during this process is fibrosis, which stiffens the heart and can cause irreversible damage. A previous study by our lab identified septin-4, a component of cellular structure, as an important regulator in this process. For my project, we hope to identify the mechanisms by which septin-4 mediates differentiation into myofibroblasts and causes fibrosis. To study this, we are staining fibroblast proteins to see which interact with septin-4 during differentiation. We are also analyzing RNA and protein expression of septin-4 compared to differentiation markers over time. Future work on this project will identify other proteins involved in this process by labeling proteins close to septin-4 during differentiation.